So when we talk about plants, there's four main groups of plants. There are non-vascular plants, seedless vascular plants, gymnosperms, and angiosperms. Non-vascular plants are the most primitive, the least complex of all the plants. But unfortunately, we don't have time to do a lab on that. We're limited in the amount of time that we have, and if there's any group that we're going to not discuss in lab, that would be the one. We will discuss them in lecture, but we're not going to get to them in lab. So I'm starting kind of with the second group, beyond that, which are the seedless vascular plants. The seedless vascular plants include things like ferns, something called lycopods, whisk ferns, which are not the same thing as true ferns, and horsetails. Okay. Now these plants are the surviving members of the earliest lineages of vascular plants. Right? They're not by any means the dominant group of plants that are out there. That's angiosperms. Flowering plants dominate because they are essentially good or best at everything. They are most competitive at doing everything. They're better at doing everything. So a lot of these types of species have gone extinct over time as they've been outcompeted by better equipped plants. Right? But these are the lineages that still survive today. Okay? And when I say vascular plants, I mean that these are plants that are going to have something called vascular tissue. Vascular tissue are essentially pipes or tubes or veins that are in plants that help them transport things like water, nutrients, and sugars that are made through photosynthesis. You've got a different set of pipes depending on what you're moving. You've got your xylem, which moves water and nutrients, essentially from the roots up to the top parts of the plant. And then you have what's called the phloem, which is transporting sugars, where photosynthesis is happening up here on the top of the plant down to the roots. Hopefully that's a little bit of review from things that you've heard in previous classes. We're going to get more into it in much more detail in the lecture, but that's mainly what I'm going to talk about right now. Obviously, the more primitive plants, the non-vascular plants, don't have that, and that's going to restrict them in terms of what they can do and how they can grow. They're not very good at moving things, and which means they have to stay small. And we'll get into that more later. But these can, and so they're going to get to be a bit bigger. Now, none of these produce any of the things that we typically associate with plants having. They don't make pollen, they don't make seeds, they don't make flowers, and they don't make fruits. So you might say, well, then how do they reproduce? All of them are going to reproduce using single-celled spores, right? Not seeds. So they're lacking a lot of things that we really expect plants to have. There's one additional thing that they are also lacking. They don't have what's called secondary growth, meaning they don't have wood. These are not going to be trees, any sort of woody plants. Most of them are not real large. There was a time period when they were. Right? During the Carboniferous period, this was the dominant group of plants that existed because the more evolved ones didn't exist yet. Right? But over time, they were outcompeted by the gymnosperms, the angiosperms that were better at doing everything. And we lost these giants that you see right here, and what we have today are the smaller versions. Now, the group that most everybody recognizes when they see the seedless vascular plants is they recognize the ferns. Right? But these other three groups you may be a little bit less familiar with unless you talked about them in 130. So I'm going to start with that group first. They used to be called the fernales, but we don't really use that term anymore. Right? They are the lycopods, the whisk ferns, and the horsetails. This, for example, is a whisk fern. This is a horsetail, also sometimes known as scouring rushes. And all of these that you see right here are types of lycopods. Right? If you look at those, they don't really look very much like a fern. And you might think, well, why in the world are these grouped together with the ferns? They look nothing like a fern. Their relationship to the ferns has nothing to do with how they grow or what they look like. There's no similarities there, or very few similarities there. Their relation to ferns is based on reproduction. The fact that they don't make seeds, pollen, flowers, fruits, and that they reproduce using spores. Okay, so that's the common feature they have that links them together, and that they have vascular tissue. Most of these, and you're going to see this today as we look at a lot of the plants, are going to have really small leaves that look almost like little scales. And we refer to those as microphyls. Matter of fact, that's actually going to be a defining feature of one of the groups up here that's called the lycopods. Right? And we'll get into that in lecture as well. Microphyls are defined not only by the fact that they're very small and highly reduced, but they only have a single strand of vascular tissue in them, which means there's only one vein in the leaf, and that's it. It's still enough, though, to make them vascular plants. They still have vascular tissue, they just don't have a lot of them in their leaves. They have one strand. So if you look at these pictures right here, these weird things that you see fused around the stem of the horsetail, those are the leaves right there. These are even smaller. You can actually feel these more than you can see them. They're like little hooks on the actual branches here. And then these little scales that you see all running along the stem there, those are the leaves for those. Now if we get into the ferns, this is the only group of seedless vascular plants that have obvious leaves that we refer to as fronds. When they're young, they tend to kind of be curled up on themselves. They look a little bit like the top of a violin, which is why we refer to them as fiddleheads. Not a technical term, but you'll hear people refer to those oftentimes. There are some species you can actually eat the fiddleheads. You can saute them up and eat them. But I'm gonna use this disclaimer multiple times during the semester. Don't go out and start eating things unless you know what you're doing. Even I don't do that. Even though I might know a plant is edible, I still don't eat it in the field. Okay? So I don't recommend that. And there's some plants out there that are highly toxic, so you've got to be careful about things like that. We'll discuss some of those families later. Now, if you look in the picture right here, we've got kind of a typical looking frond, a typical looking fern leaf. Some of these you're going to find are going to be divided into smaller and smaller segments. You've got several parts to a fern frond. If you pull up this sheet right here that I've given you, we're going to start with that one to look at some of the terms that are associated with ferns. Okay? Because most of the plants we're going to look at today are ferns. You have to know this terminology. Okay? The main photosynthetic portion of the plant, or the leaf right here, because this is one leaf. All of this right here is referred to as the blade. Okay? It may be divided, it may not be. We're going to get into that in just a second. Okay? The stalk that comes off of that is referred to as a stipe. Okay? Sometimes you'll also hear it referred to as a petiole, but usually petiole is the term we use for the stalk of a leaf of an angiosperm, not a fern. Okay? So you'll hear sometimes stipe, or you'll hear petiole. Then you'll see this one right here is actually divided. It's divided a lot. Okay? The blade is actually broken into smaller and smaller pieces. Okay? This portion right here, where all of these kind of flattened parts of the blade are located that are connected to this stem-like structure right here, is referred to as the rachis. If you're on one of the smaller branches off of here, it's referred to as a rachilla. Okay? So we've got our rachis, we've got our primary rachilla, we've got our secondary rachilla right here. Now you may have one division, you may have two divisions, you may have three divisions, it just depends. And when I say divisions, this is what I mean. Follow your stipe up here, and you can see that there's a set of branches that occurs right here. We have a set of branches that occurs right here, and we have a set of branches, and so on and so forth. Okay? This is the first division that occurs on the frond. But you can see on each of those branches, they have a second set of divisions that occur, and then even a third set, like this. It doesn't have to go that high, but it can. Okay? The very first division that you have right here, one of those pieces, is referred to as a pinna. You'll need to know that because on some of the exercises in lab, I'm going to ask you to draw the pinna. So you've got to be able to figure out what the pinna is. Right? For this one that's not really divided all that much, that primary division right there, one of those little leafy looking things, would be a pinna. But on the one next to it, which is divided more, you can see that first division includes all of this portion right here. That's the pinna. Right? Now, if we're going down to the smallest division we can find, so you can see it divides here, and then it divides like this. This is the smallest division that's complete right here. That would be referred to as a pinule. So we've got pinna, pinule. Pinna is the first division, pinule is the smallest. And we won't talk about the ones in between, just the biggest and the smallest. That's all I'll ask you in the lab. Okay, so now we can use frond division to help us distinguish the fern species. That's one of the things you're definitely going to want to look at all the ferns that you're looking at today, is what is the division here for these different ferns. Okay? If you turn this sheet over to the back, you'll see the terms that I'm looking for here. One of the things that you're going to have to pay attention to is when a fern divides, is that division complete or not complete? Now what I mean complete is that the cut goes all the way to the rachis, all the way to the stem in the center of that leaf. So if we have, for example, a fern right here, we've got only one set of divisions like this. Like something like that. But you'll notice that the cut right here doesn't go all the way to the rachis. Okay? It looks a little different than the one that's over here where it does. Right, those are actually separated from each other, whereas these are connected by tissue in between those, what they call lobes. Right? This would be called pinnatifid. And this, where the division is complete, would be called pinnate. Okay? So to kind of see what that might look like in a real fern, here are some comparisons. This one would be pinnatifid. It has a set of divisions here, but each of these lobes, if you look closely along the rachis, still has tissue that connects this lobe to this one, and this one to this one. That cut is not all the way clean to the rachis. So that's pinnatifid. Whereas for this one, it is. Okay? These leaflets that you see right here are completely separate from each other. There's no tissue in between those connecting them. So we've got pinnatifid and pinnate. But as you can see, it can get more complex than that, and most of them do that. So for example, if we have this fern, where we're actually going to have one complete division that occurs, okay, so these are actually going to be separate from each other, but the second one that happens are not complete, meaning it doesn't go all the way down to the brachilla. And we'll compare that to this one over here, which is actually going to have two complete divisions. So we've got one set that's complete here, and then the second set here is also going to be complete. Okay. This would be considered bipinnate, because it's got two sets of divisions that are complete. This one, on the other hand, only has one that's complete, so we can only say pinnate. And then the second set is not pinnatifid. Okay? And you'll see that again on the back of your sheet right here. So here's pinnate pinnatifid, bipinnate. Okay? You can go even further down here. We've got bipinnate pinnatifid. So you've got two divisions that are complete, one that's not. Or you can have tripinnate, where you have three divisions that are complete. So here's another example. Okay? This one is pinnate pinnatifid because you can see these right here are separated from each other. This is a complete division right here. But if you go to the next level right here, these are not completely separated from each other. So we only have one complete division, pinnate pinnatifid. Now, take a look at this one and tell me what the division of that one is. And I've kind of zoomed in on a section here so you can see up close what's going on with that. I'll use my little laser pointer here. Well, let's start from the basics. Do we have at least one complete division? Yes. Okay. So these right here, this one, this one, this one, this one, that's completely separated, right? We have one complete division, right? Go to the next layer. Okay? Take one of those branches right there. Look at these. Are those divisions separate from each other? Yes. Okay, so that means we have two complete divisions so far, right? Okay. Now go to the division here and look at this. Is it somewhat divided? Yes, but is it complete? No. Okay, so we have two complete divisions, one that's not. Looking at your sheet there, what would that put it in? Bipinnate pinnatifid, exactly right. So that's going to be bipinnate pinnatifid. Okay? So that's our answer down there, bipinnate pinnatifid. Okay? You're going to be asked to do that for some of the ferns today, okay? Because sometimes the division is something that's going to help you identify closely related species that look very similar. And just looking at that division will help you distinguish them. Okay? The other things that you can use to identify some of the different fern species is to look at textures that are on the stipe and the rachis. Some of them, as you're going to see, are going to look almost black or reddish colored, and they're going to be very smooth and shiny. Other ones may be covered in little hairs, like you see this one, or little scales. Classic for the Christmas fern. That's one of the first things I look for on a Christmas fern, is look for those brown scales that are on the stipe and on the rachis. Right? Other ones may have kind of an orangish color to them, or a hay color to them, a yellowish color. Right? Some of them may be colored but still smooth with no textures on them. Some of them are going to have little scales on them, right? Little dots, not these type of scales. It's a useful characteristic for several of the species to recognize, and I'll try and give you hints of that as you go through your exercises. Usually I ask you about that because it's something I think you need to focus on. So look for those stipe and rachis textures and colors. Okay. All right. Now, we need to talk a little bit about the life cycle of plants to understand some of the other things that we need to look for to recognize these different species. We're going to go into this in a lot more detail in lecture, right? But I have to give you just a taste of the life cycle of plants in lab to understand these basics. If you've had any of my classes before, you probably have gotten this before. Every plant class talks about the life cycle of plants because every plant out there, non-vascular, seedless, vascular, gymnosperms, angiosperms, has the same life cycle. It's called alternation of generations. All plants have it. That's one of the nice things about them. And it's called alternation of generations because during its lifetime, it's going to alternate between a multicellular diploid stage, diploid meaning that the cells have two sets or two copies of their chromosome sets, right? And a multicellular haploid stage, where the cells have just a single copy of each of the types of chromosomes. The multicellular haploid stage is called the gametophyte. And here's a little tip for you. They are named after what they make. So the gametophyte is going to make the gametes, either sperm or egg or both. And then you have your multicellular diploid stage, which is called the sporophyte. What do we think that one makes? Spores. Spores, exactly right. So that's going to make the spores. Now, why it's important to know the cycle is because which stage they spend most of their life in other words, what their dominant stage is, can oftentimes determine what major group of plants you're in. The non-vascular plants, for example, have the gametophyte as their dominant stage. So if you're looking at a moss, the green part of a moss, that's the haploid stage. Okay? The diploid stage is very, very minuscule and doesn't hang around for very long. But all the other groups of plants, the seedless, vascular, gymnosperms, and angiosperms, are going to have the sporophyte as their dominant stage. So when you look at a fern, the plants that you're seeing right here, they're in their sporophyte stage. There's only one station in here that's going to show you the gametophyte stage, and it's going to be right here on these microscopes. They're that small. They're about the size of the tip of my finger. Right? You can still see them without a microscope, but it's hard to do that. But that's how not dominant they are. They don't hang around for very long. So the seedless vascular plants, their dominant stage is going to be the sporophyte. We'll get that one and ingrain that one in lecture to make sure we know that. So since most of what you're going to see in the field is going to be in sporophytes, we need to talk a little bit about what the sporophytes look like and some of the features you want to look at on them to figure out what species you're looking at. Keep in mind what they make. Okay? So sporophytes make spores, gametophytes make gametes. Okay? But if you think about even human bodies, where we make gametes and things like that, we don't make it over our entire body. Our fingers aren't making gametes and things like that. We have special organs where that actually happens. And the same thing applies to plants. Whether you're in the sporophyte stage or the gametophyte stage, you're going to have special organs that are actually making these things, spores or gametes. So we're going to start first with our sporophyte. Okay? The sporophytes produce spores. The special organs that they make them in, I'll refer to as sporangia. So not the whole fern frond is going to make spores, just the sporangia on the sporophytes are going to make the spores. And there's two different types of sporangia you can find in plants. And we'll talk about this again in lecture as well. So this is one of those things that's going to cross over. You're going to get tested on it in lecture for sure, but you will get a lecture on it in lecture to refresh your memory on this. The two types of sporangia that we find in plants are something called leptosporangia Really common in seedless vascular plants, but generally speaking in plants overall, not very common. It's an exclusive group. And then the main type of sporangia you see in most plants are going to be what are called eusporangia. They are subtly different in terms of their characteristics, so let's talk about those. Okay, we're going to start with leptosporangia. Leptosporangia are found only in some of the ferns. They're actually called leptosporangia ferns. It's a clade. It's a group that is linked evolutionarily speaking. They're all related to each other, and their main defining feature is the fact that they have this specific type of sporangia. Most of the ferns that you know are going to fall in this category. The sporangia are relatively small. You actually can't see an individual sporangia without a microscope. You can see a cluster of them, okay, all grouped together, but not a single individual one. They're that small. One of the reasons being they develop from just a single cell. And this is kind of what it looks like right here. You can actually see that the wall of the sporangia right here is thin enough you can actually see the spores inside. And that's because the sporangial wall is only one cell layer thick, and that's it. One of their most unique features, though, that's something that the other group of sporangia are not going to have, is they have something called an annulus. Our terms are starting already. Yay! Right, an annulus is going to be these weird kind of square-shaped cells that you see right here that form or run in a line kind of down the back spine of the sporangia. They're kind of important because ultimately they are the dispersal mechanism. Right? They are ultimately what's going to get the spores away from the sporangia and out into the world. Right? What happens is when these cells right here dry out, they change shape and they tug on one another. Right? They kind of pull and form some tension here. And it sort of pulls it this way, almost like it's opening a cap. There are cells along here that are very weak, and when it pulls on this, when it dries out, it will actually pop that open and it will catapult the spores out away from the sporangia in the process. I'll show you a picture of it or a video of it in just a second, but I will have a link here that you can look at later if you want to see it on your own as well. Now, leptosporangia oftentimes cluster together into groups, right, which is when you usually can see where they are. They cluster together into clusters that are called sori. Sori is multiple clusters. If you're looking at just one of the clusters right here, we call it a sorus. So this is the singular, sori is the plural. And here you can see they're oftentimes found on the underside of the fern frond. Right? Not necessarily on all of the fronds or all parts of the frond, but they'll be found on the underside of it. And usually they're arranged in a very specific pattern. That is one of the things we definitely use to help identify different fern species. So you can see that sometimes they're arranged along the edges in a line, right here. Sometimes they're towards the center of the pinna, right here, and they kind of form into little V shapes that stick out. They're linear. You see some of these are in lines going away from the racula like this. It just depends. It really does depend on the species as to how they're arranged. The shape can also be important. Some of them are kind of kidney shaped. Kidney shaped means that they are shaped like this. The cluster looks like this. Or they may be more rounded or more linear. As a matter of fact, there's going to be one family in particular we're going to look at here today, which is called a Spleniaceae, that they always have these linear sori. That's one of their distinctive characteristics. So use that. Use that to help you recognize the ebony spleenworts, the only one that's going to do that. The other thing you can look for is something called an indusium. Now, an indusium is a little structure, or a little flap, that sometimes, it's not always present, covers the sori It's essentially a protective covering, protective coating, so that the sporangia don't get harmed during the process of development as they're maturing. So here you can see it looks almost like a little umbrella here. There's a little umbrella that sticks up here and it covers over top all of the little sporangia underneath this. And typically what happens is when the sporangia are finally ready to pop, and when they're ready to release their spores, the indusia will kind of dry up and sort of crinkle up and collapse on themselves, exposing the sporangia underneath them so that the spores can be easily released. If they tried to do it this way, it wouldn't get very far. So this one's not mature yet. As it's matured, it's now started to kind of collapse on itself, right, to release those sporangia. And the indusia can also be different shapes as well. So again, you can have some that are kidney shaped. Usually if the sori is kidney shaped, the indusium is going to be kidney shaped. If it's more rounded sori, then you're going to have rounded indusia. Or you can have the linear ones again, right, as an example as well. But an indusia is not always present, yes?
So those just go over top of the sori?
It goes over top of the whole sori, yep.
Okay.
Covers the whole thing. All the sporangia underneath that are going to be kind of covered by it to kind of protect them. Mm-hmm. Now, a true indusium is actually a separate anatomical structure, separate tissue from the leaf tissue. It attaches to the leaf, it sits on the leaf, but it is not leaf tissue. Okay? You can sometimes find certain types of ferns that have something that's called a false indusium, which instead of having a separate structure that covers their sori, essentially they take the tips of their leaves and curl them. And the leaf material actually covers up the sporangia and protects them. So you can see this is actually a leaf tip right here. It's just kind of rolled in on itself. And the sporangia are underneath here, but it's not a separate structure like a true indusium would be. We're only going to have one fern today that has these, that has false indusia. So that you can use to help identify it into species. Okay, all of that relates to the leptosporangia. But let's talk about the other group now, eusporangia. Most plants are going to have this. Unless you're part of that special group, the leptosporangia ferns, you're going to have eusporangia instead. And all plants are going to have this, because remember, all plants are going to have a sporophyte stage, which means they have sporangia to make spores. So we're going to find this in a lot of plants. They're relatively large, and I say relatively in the sense that a lot of them you don't have to have a microscope to see them. The very first plant sitting back there on the back corner right there is called whiskfern, Xylodium. You can actually see the little round balls that are on the tips of the branches, the little yellow balls right there that are the sporangia, without a microscope. So those are eusporangia. They develop from more than one cell, not just one, like our leptosporangia did. And you can see that we can't see through the wall here to actually see the spores inside because it's thicker than a leptosporangia was. So this one is going to be more than one cell layer thick. They also do not have an annulus, so they're going to have various other types of means of popping open to release their spores. They also do not cluster into sori. That's not a term that we use for plants that don't have leptosporangia. That doesn't mean they can't cluster. They can, but they cluster in a different way, and many different ways. We'll really talk about only one of those ways that they can cluster, but it's not going to be in a sori. Okay, one way that eusporangia can cluster is they can form into something called a strobolus. A strobolus is essentially a fancy word or fancy name for something that looks like a cone. Right? So a pine cone actually would be a type of strobolus, but there's other type of strobili besides just cones. Okay? We're going to find that in the seedless vascular plants in things like the horsetails and our lycopods. And not all of them, but some of them. There's a very generalized definition for a strobolus because it can include a lot of different anatomical features. This is the general definition for it. It is any sort of a structure that consists of sporangia-bearing structures that I'll talk about in a second that are densely grouped along a stem. Right? They do oftentimes look like a cone or a cone-like structure or a club. It's one of the reasons why we call one of the groups of lycopods club mosses is because they make these. Most of them do. So this would be an example of a strobolus, but so would this. And anatomically or structurally, they're a little bit different. Okay? The characteristic here is that we have to have sporangia-bearing structures here, all grouped together on a stem. Okay? If we go up here to one of our lycopods here, the sporangia-bearing structures in this case are actually special types of leaves. Okay? If you look closely, there's a little scale here, there's a little scale here, there's a little scale here. Those are the leaves. And at the base of every one of them, there's a sporangia. They are very closely overlapping each other. The leaves actually overlap each other like this. So we're densely grouped along the stem like we're supposed to be. And each of the leaves has the sporangia. So we meet the definition of a strobolus. But if we look down here, this is one from a horsetail. It's a little bit different looking. These aren't leaves right here. They are still structures that bear sporangia-bearing structures. They look like little umbrellas. I have these close up. These look like little umbrellas that have the sporangia kind of hanging down, but they have a lot of those all grouped together on the stem into this thing that looks kind of like a cone. So a strobili can be a lot of different things, but they all consistently fall within this definition right here. And we're going to have, most of the plants we're going to look at today don't actually form these. So I've given you some plants that are not going to be on your site recognition test that you can look at today to see what those look like, just so you're at least familiar with the terms when you see them in lecture and what they look like. Okay? All right. Now we've talked a lot about the sporophyte because that is the dominant stage. We're just briefly going to talk about the other stage because we have one station here that pertains to that. So the gametophyte is the non-dominant stage in the life cycle for seedless vascular plants. It does make the gametes, but just like the sporophyte that makes the spores in special organs, called sporangia, gametophytes are going to make the gametes in special organs too. Okay? Those special organs are called gamtangia. So sporangia, gamtangia. Gives you an idea of what they make. But there are two different types of gametes, right? There's a sperm and there's egg. So there are two different types of gamtangia. One type makes only eggs and one type makes only sperm. So we have a male gamtangia and we have a female gamtangia. The male one is called the antheridium. Antheridia can only make sperm. They can't make eggs. And then the female gamtangia is the archegonium. Can only make eggs, not sperm. Now that doesn't mean that you can't find both types of gamtangia on the same gametophyte. You can have a bisexual gametophyte. We have one right here. Okay? This one right here, if you look on the bottom side of this, these are the archegonia up here towards the top. These are making the eggs. These rounded balls down here towards the bottom under these hairs, those are the antheridia. Those are making the sperm. So this is a bisexual gametophyte because it can make both egg and sperm, right? But it has to have separate organs on it that do that. Okay? You can also have unisexual gametophytes as well, ones that are only females and only have archegonia, and ones that are only males that have only antheridia. Okay, now when the gametes, the egg and the sperm, come together and fuse in the process of fertilization through sexual reproduction, you get something called a zygote. It is a single cell that eventually is going to divide and form into the other stage in the life cycle, which is the sporophyte. That's essentially the baby plant. Right? That's going to become the fern in the life cycle of the fern. Right? So the zygote becomes the sporophyte. So you'll get a chance to look at some slides over here and kind of see that development process. You're still going to actually be able to see what the gametophyte is. This is still the gametophyte right here. Right? there's been sperm and egg that has come together and fused in the archegonia here on this gametophyte, and it started growing a sporophyte. So we have both the sporophyte and the gametophyte represented on this picture right here. Eventually this will disintegrate and this will form into the main plant. Okay. All right, questions so far? I know it's a lot of terms. Right? Hopefully you got some notes and you took good notes for this one to be prepared for your exercise. All right, now what you're going to do today, if you pull up this sheet right here where it says Seedless Vascular Plants, okay, this is your exercise for today. The purpose of the lab is to get a closer look, hands-on look, at the taxonomic characters associated with seedless vascular plants. All of the species that are listed in this exercise right here are representatives of things that you might find in Tennessee, okay, or in surrounding areas. You're going to be tested on them for your site recognition test, your first one, okay? Which means you need to know the family name, the common name, and scientific name. That will be listed for each one of the plants that are here. If you look at number 1, for example, it starts off with the scientific name, Psilotum nudum. Then in parentheses, it gives you the family name, Xylitaceae. And just a little tip, every plant family, if it is a family, will always end in the following letters. A-C-E-A-E. If it ends in something else, it's not a family. Right? So that's part of the spelling right there. If you can remember that, you got part of your family name spelled out already. So then you have, in quotation marks beyond that, the common name. Okay? Will be for that species as well. Right? The idea here is for you to kind of get a good idea of the characteristics we use to differentiate these species, especially the ones that look really similar. I've tried to put the ones that are kind of similar looking next to each other so you can compare and contrast them. You can use the characteristics I have here to help you for sure. For example, the stipe and the rachis textures and colors, the sori shape, those sorts of things. But if you see additional things on there that are useful to you, to remember that, write it down. This is your chance to take notes. Okay? You're going to have different questions that are going to be for each one of the stations. Sometimes it asks you to draw something. You do not have to be an artist in this class. Okay? I'm not grading you on your art. Okay, but the reason I have you draw it sometimes is because in order to draw something like this, you have to look at it closely, and I want you to look at these closely. That's the whole point of the exercise, is to get really up close and personal with these plants. And so that's why I'm asking you to draw. Okay? If I think there's a feature or something you're missing, I'll help you out with that drawing, make sure you've got that in there. But I'm not grading you on your drawing. That's why I'm having you draw. There'll be questions too, asking you things like what kind of sporangia are these? What is the leaf division for this plant? Does it have an indusia? Does it not? What type of indusium is it? If it has that, all those are things that can help you with the identification of the plants. Now you can start on any station that you want to. You can also work in as bigger groups as you want to. You don't have to work by yourself, but you are welcome to work by yourself. But you won't see the live specimens again until the test. Right? Feel free to take pictures with your phones. The prep specimens will stay. Right? I'll have them in a stack in a pile sitting over here on this countertop. You can come in and study these at any time. Now, one of the things that you'll notice on some of these, this will be a good example right here, is sometimes there's little tags or labels here that have some annotations on them. Okay? These specimens that right here are really old. Some of these are older than when I was born. And one of the things we're going to learn in lecture is that plant names are constantly changing. As people get new information, update them. And many of these have changed names. They're in different families, they're in different genera at this point. The ones that are on your sheet of paper right here are the ones that you are responsible for on your test, right? Not necessarily what's listed here. I've tried to update it on the annotation if I can, but this is what I want you to memorize. What I'll give you next week when we do our gymnosperm lab is I will give you a sheet of paper that has every plant that's on that first test, including these right here. But you can start studying now with this, okay? So these are the names right here I want you to know. So don't be surprised if some of the names look a little different on these labels right here. That's why. We'll talk about why they change later. Okay? You may start. If you have questions, I'm happy to come around. And when you finish all your exercises, and I encourage you to write this on a separate sheet of paper, okay, you're going to have to bring it up to me, and I have to check it to make sure it's correct before you can leave. Okay? Once I've checked it, you're welcome to leave. The reason I'm checking